All right, well, I think I'll just take care of a little bit of housekeeping. Claire should be joining us shortly, and Brandon is with us to give his presentation. So welcome to the second Lattice Lightsheet User Group meeting. Thank you all so much for being here. We're glad to have you. Agenda today, we'll just walk through some general announcements, welcome, and then Brandon will give his presentation on image analysis with Lattice Lightsheet data, and then we'll have a closing wrap up and possibly talk about next topics. We are recording these meetings so that they can be shared with people who can't attend them. So if you don't want to be seen, keep your camera off, and please use the raise hands function to ask any questions. Brandon, up to you whether you want questions during or after the presentation. And then just a quick round of introductions. Claire, I can pass this over back to you. Good to see you.
Thanks for jumping in, Vanessa. We had my Zoom kept crashing every time I tried to join, so a computer reboot seemed to fix it up. So yeah, so welcome, and so I'm Claire Brown. I'm associate professor and director of the Advanced Bioimaging Facility at McGill, and co-chairing this group with Brandon Scott, who's going to tell us all about his image analysis efforts for Lattice Lightsheet today. So we also have Nikki and Vanessa, the administrative team of Bioimaging North America, who've really been instrumental in the background, keeping our mailing list up to date, keeping everybody informed, and drafting our slides, and keeping us all on track. So we really appreciate that too.
Yes. Thanks, Claire.
So now, do you want to, you can go ahead, Vanessa.
No, sorry. I thought the next slide was mine. So there's one more after this. Sorry.
That's okay. So just to get to know each other, if you want to go ahead and put your name, your institution, and if you work in an imaging facility, the name of your facility in the chat, and then what type of light sheet you work with and what field of research you work on, so we can have a good record of who's here and what everybody's up to. So I'll just give you a minute to do that. I can tell you a little bit about another assay for live cell imaging. We were putting our new Zeiss Lattice Lightsheet through the paces, and we were doing just standard cell tracking overnight, just as a baseline, looking at focus drift, and we know our cells really well, so we know how they migrate and what they look like. And they kept dying, and we weren't sure why. We were using 0.1% laser power. And it turned out that there was a problem with the laser attenuation, and it was about 40 times brighter than we thought. And so we actually had to have the laser replaced. So we're just testing it out again this week. Hopefully everything is good now. I was a little worried that we weren't going to be able to do live imaging because there was too much power, but it was actually an issue with the attenuation. So should be good to go now.
While you guys are completing your introductions in the chat, thank you for doing that. I just wanted to share some upcoming events we have going on with BINA. So you all are here, obviously, for the Lattice Lightsheet User Group. But the expansion microscopy user group is meeting next week. If you have any interest in that topic, that will involve a presentation from Ed Boyden. And we also have a scientific speaker series and a career speaker series. So those are coming up also in later on in April, next week and the following week. So we highly encourage you to attend. And if you are attending the ABRF meeting or in Boston, we're doing a pop-up EOE hosted at the Broad Institute by Caterina Strambio de Castilla and Beth Simony. They'll be talking about image metadata. And also, of course, please save the date for our Labby x BINA Community Congress event. This is a joint event between Latin America Bioimaging and Bioimaging North America. It's going to be hosted the last week in September, and it's going to be in Mexico, but it will also be a hybrid meeting. So we hope many of you will consider joining us for many of the sessions there. Thanks, and I'll pass it over to Brandon for his presentation and stop sharing.
Excellent. Okay, so it's going to be, so hopefully this works out okay. Unfortunately, we have a blizzard here in South Dakota, and so we had a foot of snow overnight. And so I'm not able to get to my work computer, but I'm going to remote in and be on Zoom in both places. So we'll hope that it works. I tested it last night and it worked okay. But if it's, less awesome than normal, it's not me this time, I suppose. But we'll see. So I'm going to try and get on there and share the proper screen here. Okay. So we'll share, hopefully. Sorry, give me one second here. We'll get there. Okay, can you all see my presentation now?
Yes, we see the first slide of your presentation.
Okay, excellent. And so I would say, please, I actually can't see your hands or anything. So if you want to, I would very much say, let's have this be interactive and ask questions along the way. I'm going to get that out of the way. This is just an attempt at, showing some of the different things that we're doing in my lab related to image analysis. So it walks us through from the start, a little bit in terms of how we get from the raw data to the finished product. And then I would like to try and show some of the things that we do in Chimera. This is the part that might be streamlined than it might be, but we'll do there. So as we get started here, I think this was a very nice schematic from this paper from Michael Weber, really showing the multitude of disciplines that are needed. And it's really this interconnected nature of what you need to do, an image experiment well. And that really comes down to the same thing in terms of what is needed for what I think an imaging scientist is, and that is really that an imaging scientist needs to be this man, bear, pig, right? Where you're half man, half bear, half pig. So you can do everything all at once as well as you can in here, okay? And so everybody knows this, but I just want to show what it looks like as we get some data off of the Lattice Lightsheet microscope here. So some raw data. And the question really is, maybe for people who are new or not as well versed in this, is these aren't exactly helpful slices to be looking at here in terms of this raw data. And so we have to take it from the raw data to something that is usable. Okay? And so this, again, maybe for this group, we don't need to talk about it, but we're going to, right? As we're scanning through, we need to de-skew this data. And this is the first step that is a major bottleneck and took quite a bit of time to get into a streamlined process. And where we are now is it works well, but it's a little, there's still a lot of manual input in there. And so here, right, this is nothing that we did. We would be nowhere without Tally Lambert, right? I think in terms of where my lab is at all, right? We use so many things that he's developed, and it's been an amazing resource for us. So I just want to give him a shout out here because it seems like everything that he touches is just gold, right? And so we use a lot of that, and I want to give credit where it's due. Okay, and so with that, we can take and use basically his little LLS SPY GUI to do this pre-processing pipeline. The biggest part that we end up having, and I think everybody has, is there's a lot of transfer steps. So the microscopes, their hard drives have a finite capacity, and the analysis workstations are typically in a different place. And so the first step for all of these is transfer from the microscope, which I'm labeling S, to some storage drive, which in our case is like 150 or 200 terabytes of external storage in a big RAID array. And so from there, these are slow though. These drives are slow because they're for long-term storage. And so then we have to copy it from, that storage to a local NVMe drive, which is very fast, but has a smaller capacity. So this is on a, per experiment run, we have to do these steps. Then on there, we have to, in our case, go from multiple packages. So from Python using LSPy into MATLAB to crop it, back into Python to compress it, which is how we like to store it longer term. And then we're ready to do something with that. And then once we've done that, our drives are too small, and so they have to be moved from this fast drive back to our long-term storage. And we are basically storing just the 3 parts, right? So we're storing the MIP that's regular, and then we store the volumetric data that's been cropped. So in this step, as most people do, it's this deskewing and some light deconvolution on the data. And then, so we're saving that data and then the crop down region that we're actually interested in. And so this takes a lot of work to get there. And so one of the things that we've done is developed a more streamlined pipeline. And I think others, we're certainly not the first to do this. This is just the way that we're doing it, where we have an automated transfer from our microscope to our NVMe drive all at once. And then we rewrote some things, getting rid of the MATLAB to Python transfer, so we can just stay all in one environment. And then once this is all completed, then we move the data, both the original and the pre-processed, from this fast drive back to our storage drive. Okay, and this is all done automatically. And so that's really helped to streamline this process on a per experiment basis, where you basically, if everything is the same that you would use in this GUI, then you can just run it once and then come back the next morning and you have all of your data that has been pre-processed. Okay. Okay, and so I would say, you know, as again, I just want to make a point, questions, comments, anything, please. If others have things that they're like, oh, we do this and it works really, really well, now would be the time to, you know, shoot a hand up and jump in. So, okay. Right, so what does that look like? Here's now a movie that we've rendered as in 2 ways, showing a target cell that is going to be internalized by our macrophage. And in this is all done now in Chimera, which is where we do, this is what we're going to talk about for a lot of, for the rest of it, is different ways to visualize the data first, and then our efforts to quantify those data. Because I think, you know, talking about how data is big and we need more storage, everybody knows that. Let's try and figure out how do we get to the next step in here. And so what we can do in these is really try and show different ways to visualize the data, because I think we can learn a lot just by even looking at this data. And so in this case, this is a combination of methods to show the data. So we're showing both a volumetric MIP in that magenta color here of our targets, and then this wireframe mesh. So using this isosurface to tell us about the structure of our macrophage with respect to how it's eating that target. Okay, and this can be a very powerful way to view the data. And so we're working on a publication that is a user's guide, essentially, to doing volumetric visualization analysis in Chimera, a very straightforward way. And this is, you know, our flow diagram in terms of how do we get from the start to the end. And so the way we tend to think about it is first use a MIP, because that seems to be what everybody's most comfortable with using, and you can do it very quickly. And so you're like, okay, does the data look good? Is it time to move on? And these are really the different things that we're trying to do. So if it's looking at the surface structure or depth with respect to 2 different objects, then you might look at these isosurfaces. And then if not, then we can use in the same piece of software, we can then say, well, let's look at all of the volumetric intensities all at once, and then start to combine these different modalities into either one view, or in different subregions, we can start to look at different bits and pieces in terms of what it is that we're actually interested in doing. Okay. And so here would be an example now of a movie showing the different capabilities that we can do. So this is split up into thirds, so we can look at colocalization between two fluorescent proteins here. These are two volumetric intensity projections, just an isosurface rendering of our macrophage, and then this combination of the mesh and the volumetric intensity underneath of that. And so in each one of these renderings, as this is playing through, we get different pieces of information, and it's really incumbent on us to figure out what's the right one to use at the end of the day. And so in this case, we want to see, the structure of the membrane, but also the underlying signaling proteins that are associated with that membrane. Here, we want just a very clean view of the membrane surface. And then this one, we lack all of the structural information between them, but it enables us to start to look at colocalization between those different molecules. Okay. And so, getting beyond that is where it becomes really challenging. So this is a very simple experiment in the lab here, where we're trying to understand the interactions of these diffraction limited spots that are representing antibodies here on the surface of a cell, a signaling molecule, and then the structure of the macrophage as it's eating it. And so the experiment is quite easy, but the analysis on here tends to be quite difficult, right? So we're trying to track these surfaces, track the intensity profiles over time for both of these molecules, and correlate those three things in space and time. And so this is where most of the data end up looking like, where we're just, take a montage and you're like, yes, we can see where it is, and start to visually see how it's changing over time. And so that's like the next step is really getting to this point where it becomes a very computationally intense problem to try and solve. And that's where we're really working to move towards. So to do that, in there, we can take advantage of some of the work that's been done by others to look at the local intensity around this triangular mesh, right? So that, this is the processing pipeline here, is you threshold to make that triangular mesh, which is the same as the isosurface. We can then draw circles essentially around each vertex here and integrate the intensity that's relative to that sphere, and then color the surface with that. And so this idea is not ours, but what we've worked on is making this computationally tractable. And so if we look up here, where for a single time point, the state of the art was really 950 seconds to do one time point. If you think about what it is for movies that are 100 to 150 frames long, that would take 26 hours for us to finish those data. Just the analysis of one movie would take a day. And so I spent quite a bit of time to figure out the bottlenecks here and eliminate almost all of them. So now, those the same exact movies, instead of taking a day, now we can do in less than 5 minutes. And this, again, is all within a bundle that I wrote for Chimera that runs inside of the native Python shell that's in there. So the Chimera is all built on Python, so it uses the same ecosystem as to be able to do both of these things all at once. And so what does that look like then? So now if we can start to show our surface renderings in a different way, this is now quantitative, but getting to the point of a graph is actually still quite difficult, because we're still left with the fact that we have a four-dimensional object, 3D over time, and trying to show that as a plot becomes very challenging, because what is the question that you're trying to ask? Are you trying to say, this spot here, what does its intensity look like over time? Well, you can visualize where that goes, but from here to here, where did it go? And so at this point, it's just like, this is as good as we can do right now, and is an open area for some improvement by others, I would say. But at the end of the day, we now get to the point where we can see these surfaces colored with either, with two separate molecules, and look at the clustering and the recruitment of these downstream signaling molecules with respect to the target of interest. Okay. Okay. And so for me, that's super cool. But that's not the end, right? What I really want is others to be able to look at this data and interact with it on their own. And so these isosurfaces, the nice thing about that is we can export those as a GLB. So this is a binary GLTF file, which is a standard 3D file format that you can then use in web applications, which is really where I'm really interested in taking this. And so the problem with that is this single frame is about 18 megabytes. Now the raw data coming out of the microscope is 100 megabytes per time point. So this is already a reduction. But when we think about the web, megabytes or hundreds of megabytes is quite large. And for all of those 100 files, now we're up to almost 2 gigabytes for these isosurfaces, which you would not be able to render on the web and have those be interactive. But what we can do is make a perceptually indistinguishable object. So I think if you were to look at these two even very closely, you would have a very difficult time telling me which one's which. But our file size tells us a very different story. We're down into 500 kilobytes for this rather than 18 megabytes for that. And that really shows up when you have all 100 files. Now we're down into the 46 megabyte range, which is very, very doable for a web application. Okay, and so that is what is shown here, which is going to be, so this is a web viewer that we have built here that you're free to go and visit this one, and you can interact, and it'll look much better on your local machine than it will through Zoom, through PowerPoint, through the web, and all of those layers. But even from here, I think it still does a very nice job to show some of the capabilities that we're building in here. So we have full interactivity between here, and if you had a mouse that could zoom, you could, and you can pick basically any point in time that you want, and these are going to be displayed to you in real time. And you can also do this on a cell phone, which makes getting our data from just the clusters and the HPCs into the hands of people who are most interested in seeing it. So is that all? I don't think so. I think we have some here, right? These files, the way in which we're saving data, and this is a big issue for me. So as we're looking at these four, again, I would challenge you to try and pick which one is the largest file. And I think by eye, you could not do it. So these are not analysis things at this point, because they are lossy in their compression. You can tune it, but we can't do any more work with them in terms of trying to analyze the underlying data. But at this step, we're not trying to do that. We're trying to visualize these data in different ways. And so if we're smart and we use the right compressions, we can take what ends up being, this is a TIFF that's 8-bit. And so you can see the size is in kilobytes. So we can show where we end up. And here, this one is a single WebP format. And so you can see this dramatic, 400 times smaller if we go from this last one, which is the animated file that we can show here. And so now this full volumetric data becomes 391 kilobytes, okay, for a single time point, which the original was, 200, and this is the raw original was like 120 megabytes. When it's been at 16-bit, when it is deconvolved and we need to upsample it, it becomes half a gigabyte or something stupid like that. And so now we're down to less than 400 kilobytes. So why do I care about that? Well, what we can do is something that I think is very, very exciting. And in here, we're working to build a volumetric viewer for full volumetric data, full volumetric movies. And so again, if I had the right mouse, I could zoom in and show you these data up close, but these are full, the intensity values that you can slide and change their view in real time, again, on something as stupid as a cell phone. And so that's really where we are pushing this towards, is to get from the microscope through some complicated analysis and then out into a way that is easily shareable with others. So, okay, so that's kind of my spiel on what we're doing. I'd love to take some questions if there were any, and then I can show some of the things that we're doing in Chimera. So I'm gonna pop over to this other one so I can see it a little clearer. So I'm gonna stop my sharing for just a second here and come back here. So we'll open it up for questions and conversations first, and then we can show some of the Chimera things. If I haven't bored you, obviously.
A question just sort of from the practical point of view, like my facility is mostly biological users, so they're not using different platforms, mostly commercial packages or J. So if somebody wanted to set this Chimera up, is it a standalone package, or are you running it on, like a Python interface, or what would that look like?
Yep. So the install of Chimera is DoubleClick. It's an installer. So it's on any, you can do Mac, Windows, Linux, whatever you want to do, and then it's fully for Windows is an exe. And so the interface with that is, which I will show, is very straightforward in its usage. The learning curve is like very low, I think. So for people who are not well versed in coding, and most of my students are not well versed in coding at all, and they are very easy to pick that up.
And then is it fairly good at opening different file formats?
So I mean, we tend to stick with TIFFs going in. It'll open any other image format. We haven't played around too much with it. I mean, in terms of opening the others, that's the file format we use. Right. So TIFF in, and then we end up saving these GLTFs out is the end.
So you need to put in some of the metadata, I guess.
In terms of
pixel size, time scale, things like that.
Right. So there becomes a question of where at the end you go, right? And so if you're making some of these movies with timestamps, we end up actually doing those in ImageJ. So the metadata is kept through with the images, but yeah, they don't really play a huge role in terms of the need in Chimera, right? Yeah.
Thanks.
Sure.
I have a couple questions. First off, a really, really interesting use of Chimera. I know about it from working with molecular data primarily.
Yep.
So it's interesting to see, this data type used with Chimera. If you have the time, it would be interesting to hear your overall path towards picking Chimera as a choice for this problem. But that's not really my question. My question actually was, once you have done all of this compression and optimization for a web file, that's just the file. Yes. The promise of web is a lot of interactivity and, user feedback, but when you just have the file in hand, that's only a part of that. You would still need to design a website or use some template to work with those data. Is that part of your quote unquote solution here, or is that something that the end user would have to kind of customize?
Sure. So I will drop this in chat. So again, yeah. So this is very much things done in my lab, done by me actually. And so these are not a commercial solution by any means. But I aim them out that others formats is very to hop in and for these. So I'm preparing to make them very low in terms, and so
Getting a pretty poor connection with Brandon. Is it just me?
No, I think it's general.
You might try turning your video off, Brandon. Your feed is really cutting out.
Is that better?
Sounds like it, yes. Thank you.
Yes, we can hear you much better now. Thank you.
So we couldn't hear your answer.
Right. So in this case, the viewer itself, to add your own data to it and make your own viewer is simply uploading your file and hosting it in a free website, right? That is what Glitch is. So it's a development web. So
that's really cool. I'll definitely be playing around with this. Just to play devil's advocate, I suppose. I don't know how else to put it. Being from a commercial team, we have a sort of solution for this, but it takes a completely different spin on it, which is a preference towards keeping the original data set quality and hosting that and rendering it from a server. So it's more of a streaming technology rather than a compression technology. If anyone's interested, they can reach out to me. I'm not even mentioning the product name or anything, but it's just interesting to see a different approach with pretty good results. They look pretty nice.
So I would ask a question there. So you're streaming their just static views, right? That then the user is able to rotate.
No. It's if you've ever used any 3D software, I mean, it would feel a lot like that delivered through a browser. It's more of a toolkit rather than a standalone application. So end users design their own thing, much like you've done.
Okay. I mean, I would be very interested in learning more about that. I think this was for me. I looked for a long time to try and find something, and was unable to find something that was satisfactory. So that I set out to go to.
I'll email you about it, and
yeah.
We're not the only player in this space, so I can even alert you to some other tools.
Yep.
Blair had put in the chat. I don't know whether anybody else wants to add to the chat, but was asking about
Yep.
software packages generally.
Yeah, and I see the plugin there for Agave. So yeah, the data look beautiful in there. I will give them that. Claire's question about the barrier to entry one was for me, it was quite high. And so it was like, it looks great, but I don't know how to make it work. So to answer the question, how did we land on Chimera? Well, we landed on there because we started when we first were building the Lattice. We started with I can't remember it. I scrubbed its name from my memory. One of the commercial ones that was like 20K or something. It's now a 20,000 digital paperweight for us because it took overnight to load in data into this thing. Sorry, just knows the name of it. And so again, we couldn't find anything that worked. And then we had seen a paper that came out from UCSF that used Chimera initially. And we're like, that looks great. And we tried it and it was. So it's kind of been a no turning back once we found one that stuck. And we couldn't beat the entry price as well. So, right, I mean, they had all of the sweat equity in for us. There it was, Ariba. Yeah, I knew somebody else knew it.
I don't know, maybe if there's any other questions, I'll let anyone else go first. So I was just thinking, based on the conversation so far, I wonder whether that's something this group could do together, and maybe as a resource for the community, is maybe we could have, we have a lot of commercial representatives on the group, and there's probably others using open source and other platforms. I wonder whether we could have a series of these types of presentations with the idea of showing sort of the things that the software is really good at, and then maybe some of the limitations, like it looks like Chimera is going to be really good for visualization and stuff, but maybe if you then want to start measuring intensities, maybe it's not the best solution, and maybe we could, as a group, maybe at the next session, we could have a couple of 20-minute presentations from a couple of the companies of their solutions, obviously in a very academic sort of presentation, of how things are designed, what's going well, maybe where some of the bottlenecks are, and then we could maybe even draft up a document of sort of, or a table, if you want to do this is the software. Or I think we could also add in the user interface and what's the learning curve and things like that. What would people think of that as a way forward?
Yeah, I mean, I think if people are interested in that, I think that's a great idea.
It seems like I keep thinking with these user groups, I want it to be useful for everybody,
Great. Yes, absolutely.
I know we're just getting into things ourselves and haven't done a lot of playing around, but like, for instance, we could very quickly try Chimera and see if we can get it to open the Zeiss files, and then we could compare it to the Arivas that we'll be using with the Zeiss system, and are there things like you say, for putting it up on the web, maybe the Chimera is a really, really good solution. And if you want to track vesicles within an object and have the surface and the vesicle intensities, maybe there's something else,
Right. Yeah, and I think in there, I think those would be great if the vendors wanted to give their non sales spiel.
Yeah, for sure. Yeah, and I think it'd be interesting to see. I'm sure that the companies also know where they need more development, and that's also where we can probably help in that direction too, of sort of where we see the real needs of the community and what are the gaps that need to be filled.
Yep.
So maybe if any of our corporate partners are interested in giving a 20-minute overview at our June session, you could just send a note to Brandon or myself, Brian, and we could make up a schedule for the next few meetings to do that, with the goal of coming up with a bit of a bit of a review and comparison table that people could agree on, you know?
Yeah, so do you want to see any of what I would call the demo for Chimera, some of the interaction for that? Would that be useful to people?
I think so. We still have another 15 minutes. So.
Yeah, okay, excellent. Okay, so I'll pop back over to here, and then hopefully, let's see, we can share this again. Okay, so sorry, my desktop is an absolute nightmare. Okay, so you're seeing this now. Okay, so let me pull open Chimera wherever you went. Okay, so in here, the way in which why I think it works as well as it does for us, anyway, is using the barrier is quite low, and so it's a lot of actually drag and drop, and then we have a series of these kind of command files. You could run these all at once in a command file, but if you were to just copy and paste this to open up some data, we can do that very quickly. So if we open a new piece of data here, I've done some analysis so I'm going to keep them, but I'll add a new one to show basically why we do this, and that is how fast this is. So we've loaded in a series of data, and now we're going to pick our threshold, and we'll zip through some data here. I would very much challenge. This would be throwing down the gauntlet, if you will, in terms of speed from time to open to interactivity. Is going to be unmatched. So that was opening the series of TIFF files and generating isosurfaces, and that was basically faster than I can click and sloppily click between them. So if we try and do that again to show here, opening those guys up, and you can see it's like happening near instantly as we now select any of these other views that we want. So the question as to why did we go from here? Well, I didn't have to wait a day to load in a proprietary file format and then have a surface rendering that did not do justice to the data that I had taken. And so if we were to then go and bring back, so in this case, we have a nice model panel here where we can start to pick and choose different models that we're showing, and have both volumetric data. So this is this volumetric projection that we can see our antibody surfaces here. We can mix and match that with different isosurfaces and have multiple isosurfaces at the same time. And again, being able to play these through, and now I'm on the wrong one. So this is to say it's not without its quirks, right? Obviously, there are some issues, and I think I probably broke all of my hard work by trying to show another piece in here. Let me get this map series to be the right one. Damn it. Where did you go? All right, we'll start again. So you guys can see it from the absolute start, I suppose. All right. Okay, so let's close here, and now we'll go ahead and load in these data. And I'll do it a slightly different way here for these others, right? If we were to just copy and paste in the file that we're trying to look at And the nice thing about this is we can have these wildcards in terms of which which of the stacks we want to load in. If we want to load in all of them, we can basically just tell it to, I think, open all the files here, have all of those open up, and I'm just going to clean them up manually. This way, actually showing here, we're basically setting our surface intensity level. And so this is what dictates that surface threshold for the three different options here. And so the nice thing about it being able to copy and paste in there is it becomes a bit more reproducible in that way. So once you get it to a point that looks reasonable to you, you can make that same visualization again. And then here we're going to be loading in all of these 3 time points, which has the full volumetric data associated with these. And so the first render in this one is going to be a little bit slower, but then the next time it plays through, once it's been properly cached, it'll actually play back faster than Zoom will stream it. But if you were looking at it on your machine locally, it would play back very smoothly. It is not this jerky notion that you see, again, through this mangled mess. So from here, if we go back and play those initial ones, which have been cached, that'll show you what it looks like after that first time through. Okay, and so these are now, again, there's lots of noise that you're seeing in here associated with the data itself. And so we can start to have different ways to clean these up in terms of the surfacing and getting rid of some of that dust. So let me do that. Oh. So on here, should be able to have this go. And sorry, I was pulling this together. It turned out to be closer to last night. And so this isn't as polished I would maybe like it. But at this stage, we should get rid of all of these very small dust things here. Basically, you're looking for connected components that are less than 20 or 5, depending on the different surfaces. And so we're working on, this big gray surface and the green surface in one instance, and then we're looking at the blue surface in the second instance. So you can start to mix and match what you're doing between those. And so if this looks very clean, and we have these nice shadowing. So the beauty of it being in this crystallography and that realm for molecules is they've done an amazing job in terms of making the ray tracing and the lighting and everything make the data pop out. And so from here with that shadowing, you really get a sense for that this ruffle is coming out. Whereas if we made, say, the lighting flat, right, that completely goes away. And so there's you have full control over the way the data are presented in that way. So let me see. Okay. So then we did work out some of these analyses in here. And so, from the standpoint of being able to visualize these data, so if we change this one over to a maximum projection, we can then start to show these data as I was describing before. So looking now at the full volumetric data and being able to have control over the lookup table that you're applying to it, changing those thresholds for the way in which they are being shown, and then trying to get to this point where we're, let's see, we measure the distance from like number 1.1, which would be the first model, to our second. So if we go back to this first one and we say we want to measure it now from 1 to 1.2, and we're going to color that surface based on how much that surface moves. So this would be the Euclidean distance of that surface in the next frame. And so we have the opportunity then to say, do that for all of the frames. So if we said from one to, I think there's 50 frames total. So if we did 1 to 49, and then said from 2 to 50, that will go through each frame and measure the distance of all of the vertices with respect to one another over that time step. And so with there, we, the little spinning beach ball tells us that we're working, and this will only take, this should not take very long, except for the fact that I failed to pre-render all of them. And so my apologies in terms of loading all the data in first. So there's an order of operations that we need to use. And if you don't, well, you suffer this pain. But yeah, I mean, this is just some of the things in terms of how people interact with it is these very simple commands of say, measure something, right? Do something. And the user guide for this is going to be kind of your best friend in all of these things. That's another big selling point for us is the community of developers that are doing this work. So let me change this back. Yeah, questions, comments?
I have a question.
Yes, please.
So it sounds like you use the built-in measurement capability of the software. I'm wondering, so if you export any of these meshes or surfaces, can you, I mean, are they binary files? Can you analyze them in something else using another Python module or something like that?
Sure, that's a great question. Actually, this one's not built in. I wrote this as a package into Chimera. And so, yes, all of the data, so we basically just have it say, so each vertex has an associated intensity, and we put multiple metrics on each vertex, depending on if it's this intensity or the distance that the vertex traveled. But the nice thing is, it looks like it was built into Chimera. And so there isn't any, the need to go out. You can add additional capabilities. So this is the measure distance, that and the measure intensity. Those are two things that I wrote to interface with Chimera. So yes, you could absolutely export those any way in which you want.
That's cool. So I guess when you download Chimera, you'd have to also download the code that you wrote.
So there's the tool shed in Chimera, and so there are others. So in here, try and see where it's at. So in like tools, forget exactly where it's at, but there's a tool shed, maybe under more tools. I don't remember. I have to look. That you can basically, it's like plugins that you would have for ImageJ.
So I think we better wrap up there so we stay on schedule. But thank you very much, Brandon, for the information and the mostly flawless demo.
Oh, I guess see my office.
Probably didn't see this in the chat, but Alex wrote that they call it the demo effect. So when things don't work during the demo.
Oh, of course, yeah.
You have no audience, everything works well, right?
Well, and I'm happy that it worked as well as it did, given the hoops that we're actually jumping through to do this. So
yeah. I know, I think it gave us a really good appreciation of what you're doing and the capabilities. So Just a note that our next session will be in 2 months on Tuesday, June 6th at this same time, 12 Eastern, 11 Central, 10 Mountain, 9 Pacific. I feel like a radio announcer. And as we mentioned, if anyone representing corporate groups who have software you'd like to give us some highlights, it'd be great to maybe do 2 at the next session, and then continue that for as long as we have new options to explore.
Yeah, and
was
I
there
would also open it up to other, academic
for
groups
sure.
as well, right?
Good
I mean,
point.
others are probably
Yep.
doing this better than we are, and so I would love to hear how you guys are doing it. So this is just one way of doing this. So
And Apari would be another one. Would be
of course,
interesting.
yeah, and
Mm.
we've tried. We've tried. I went through countless, and so I would love to see somebody use it that actually makes it work.
Mhm.
So.
Yeah, and I think we can focus on the pros and cons, because I think no package, as we know, no software does everything we want. There's always one that does one thing better and another does another thing better. So I think we can really focus on teasing that out a little bit for the community.
Yep.
Mm-hmm. Excellent. Was there anything else, Vanessa, before we wrap up? We're good. All right. So thank you everyone for being here, and we look forward to seeing you in 2 months. And don't forget to sign up for all those other BINA events and save the date for the annual meeting.